Modern infectious disease therapy utilizes antibiotics as precision molecular tools, each engineered to exploit a specific biological vulnerability. Relying on rote memorization of trade names or broad drug classes breaks down in clinical practice. The variables of patient biology and pathogen resistance are simply too complex for a flashcard approach. Instead, the drug must be mapped directly to the pathogen. We match the antibiotic's mechanism of action to the exact physical anatomy of the bacteria. This gives us 4 foundational targets that organize the entire antibiotic arsenal: the external cell wall, the internal ribosomes, the coiled nucleoid, and the inner cell membrane. Prokaryotic bacteria construct these cellular structures differently than human host cells do. That biological divergence allows for selective toxicity. It allows us to achieve the primary clinical objective: eradicating the invading pathogen while minimizing systemic damage to the patient. By organizing antibiotics by their structural targets, a clinician can logically deduce a drug's utility, its limitations, and its potential for resistance without ever relying on memorization. We start at the outermost perimeter of the bacterial cell, the rigid peptidoglycan matrix, or cell wall. The primary disruptors of this matrix belong to the beta-lactam class, which includes penicillins, cephalosporins, carbapenems, and monobactams. This diagram shows those 4 drug subtypes. Despite differences, they share the exact same core architecture, a 4-member beta-lactam ring. Look at the acyl D-ala precursor molecule in the center, a natural bacterial peptide. The beta-lactam ring mimics its physical shape. Because of this mimicry, the drug tightly binds to and inactivates the bacterial enzymes responsible for building the wall, known as penicillin-binding proteins. Cross-linking halts completely, and the entire cell dies. In response, bacteria had evolved a direct countermeasure. They secrete beta-lactamase enzymes that physically cleave that critical 4-member ring, rendering the drug entirely useless. Clinically, beta-lactams act as time-dependent bactericidal killers. To be effective, the free drug concentration in the patient's body must remain above the minimum inhibitory concentration, or MIC. This chart visualizes that parameter. The duration spent above that threshold dictates the drug's lethality. If the free concentration dips too low, the bacteria survive. This strict pharmacodynamic requirement dictates why beta-lactams are often administered through continuous IV infusions or very frequent redosing. Vancomycin, a glycopeptide, provides an alternative way to inhibit the cell wall, but it comes with a severe structural limitation regarding gram-negative bacteria. Gram-negative cells possess an additional outer membrane equipped with narrow porin channels. Vancomycin is simply too massive to physically fit through those channels, restricting its use entirely to gram-positive infections. The physical shape and the specific pharmacodynamic curve of a cell wall inhibitor entirely dictate its required dose, its frequency, and the precise spectrum of bacteria it can kill. Now, moving deep into the bacterial cytoplasm, we target the 70S ribosome, the engine where mRNA is translated into functional proteins. This schematic maps the protein synthesis process. The bacterial ribosome is divided into 2 distinct functional components: the smaller 30S and larger 50S subunits. Both are highly specific targets. Looking at the bottom section, aminoglycosides and tetracyclines bind directly to the 30S subunit. Aminoglycosides tightly bind the ribosome and scramble the translation process, causing severe misreading of the mRNA codon. The bacteria produces non-functional proteins, triggering rapid cell death. But to reach that ribosome, aminoglycosides face a crucial physiological barrier. They require an oxygen-dependent active transport mechanism just to cross the bacterial cell membrane. The clinical consequence of this transport requirement is absolute. Because they need oxygen to enter the cell, aminoglycosides are useless against anaerobic bacteria. Shifting focus to the top half of the structure, we find inhibitors targeting the 50S subunit, such as macrolides, clindamycin, and linezolid. Macrolides work by physically plugging the exit tunnel on the 50S subunit. This blocks the newly born polypeptide and completely halts peptide chain elongation. Because macrolides, clindamycin, and chloramphenicol all share overlapping binding sites on this tunnel, combining them in a patient causes antagonistic interference as the drugs compete for the exact same physical space. By specifically exploiting the structural differences between the prokaryotic 70S ribosome and the eukaryotic 80S human ribosome, these drugs achieve powerful antibacterial effects while largely sparing the patient's own cellular machinery. Further inside the cell is the nucleoid, the region housing the tangled, supercoiled bacterial DNA. The premier agents for disrupting this genetic core are the fluoroquinolones, such as ciprofloxacin. During replication, bacterial DNA must be rapidly uncoiled, copied, and then tightly rewound. Fluoroquinolones specifically target the enzymes executing this mechanical process, DNA gyrase and topomerase 4. This inhibition paralyzes the replication machinery, causing the DNA strands to snap. While cell wall inhibitors require the bacteria to attempt active growth to be effective, this mechanical failure provides an immediate direct kill. There is also a secondary pathway for stopping nucleic acid production, achieved by starving the cell of its base metabolic precursors. A combination therapy like sulfamethoxazole trimethoprim sequentially blocks the synthesis of folate. Without folate, the bacteria are entirely deprived of the purines necessary to build new DNA strands. Targeting the genetic blueprint results in a high-speed bactericidal effect, but it carries a significant cost. Because our human mitochondria share an evolutionary ancestry with bacteria, these drugs can induce oxidative damage in our own mammalian cells, leading to the serious side effect warnings that now restrict their clinical use. Our final target rests just beneath the cell wall, the cytoplasmic inner lipid bilayer. Agents like daptomycin act as highly specialized, heavy-duty membrane disruptors. Daptomycin uses a unique calcium-dependent binding mechanism that allows the massive lipopeptide molecule to insert directly into the bacterial membrane. Once embedded, the drug instantly creates channels that trigger a massive efflux of potassium ions out of the cell. This sudden loss of ions forces the membrane to depolarize. All intracellular synthesis shuts down, leading to rapid cell death without actually blowing the physical cell wall apart. Much like vancomycin, daptomycin lacks gram-negative coverage due to the outer membrane barrier. A drug's physical access to a target is just as critical as its molecular lethality. Zooming out from a single cell, we have to look at the ecological scale of a patient's natural microbiome. The harsh clinical reality of pharmacology is that no single antibiotic kills absolutely every bacterium in a given population. In a healthy state, normal flora actively compete for essential nutrients and secrete inhibitory substances. This keeps dangerous mutant bacteria and opportunistic pathogens firmly in check. This chart tracks exactly what happens during broad-spectrum therapy. The green dashed line, representing the sensitive population, plunges toward zero. This mass eradication creates a biological vacuum with zero competition. Tracing the red line, resistant mutants exponentially climb over 25 hours, becoming the dominant bacteria. The clinical manifestations of this exact curve include the rise of MRSA, multi-drug-resistant gram-negative rods, and aggressive C. difficile superinfections. Utilizing an overly broad-spectrum antibiotic is an evolutionary pressure that directly constructs a far deadlier secondary infection. The ultimate mandate of the prescribing clinician is to use the anatomical vulnerability map to select the absolute narrowest spectrum agent possible. The goal is to eradicate the immediate threat while preserving the delicate, life-saving ecology of the host.